I see we have lots of questions coming into our Q&A box. So if you have any, please pop them in there now and we'll get to them in a second. As I mentioned earlier, we've also Kevin Connolly from the Food Standards Agency joining us, and Kevin will help us address any specific questions from Northern Ireland food businesses. So I'll open the questions up to the panel unless there is a specific question directed towards a specific speaker. So if everyone's happy with that, I'll get started. So our first question is, for a small food business, what examples would you recommend for gathering physicochemical data?
I'll take that, Mary. I'm not sure what they mean by examples, but maybe how would you gather?
Yeah.
So I suppose it depends on your product. So for example, pH meter, I think a lot of food businesses might be able to purchase a pH meter themselves and be trained on how to use that and just make sure they calibrate that properly, and they could test the pH at the end of every batch if they wanted and build up that profile. For water activity, it's a little more tricky. So you could buy a water activity meter in-house and be trained how to use that, but I believe it's a little bit more tricky than a pH meter. So maybe you could send samples to a lot of the commercial labs will do physiochemical tests as well as microbiological tests or chemical tests. I think there are two good ones to start with, but also I suppose looking at your product itself, if you're using a preservative, some of the models will allow you put in use of a preservative and you can see that effect, but of course you need to comply with any legislation if there's upper limits for the use of particular preservatives such as nitrate. I don't know, Catherine, if you want to add anything to that.
Yeah, no, I think you're right, Mary. I think the most labs now will give pH and water activity as standard if you even want to do a basic nutritional to cover off your salt and your preservatives as well. But while it may be costly, it's certainly it's the easier option, I would think, to send it externally.
Okay, thank you. And just following on for that, someone's asking if it's an issue if a food business isn't using pH and water activity as part of shelf life studies or routine monitoring.
Well, I suppose in terms of the more holistic approach to shelf life studies, not just, I suppose, determining shelf life just for Listeria monocytogenes, it is advisable for food businesses and best practice to understand their product in terms of the pH and the water activity and just really understanding how pathogens might grow in that product and what the profile is. I suppose it's not like the for Listeria monocytogenes, I would say it's critical to understand that, but depending on the product, maybe you might decide it's more, critical to do some other testing in terms of maybe the salt content, whatever. I think it's very case by case, depends on the product. And if you do a little bit of research to understand your product and what are the things that you need to kind of characterize. And that guidance as well, the EU guidance is a good resource to read around that.
Perfect. Thank you, Mary. So the same person has asked this question then. Will there be any testing done on products between leaving the factory and the end of shelf life, i.e. the monitoring gap?
Okay, so in terms of official controls, a certain amount of samples are tested under official controls in Ireland. So obviously not every sample is tested, but they do it on a risk basis. So and every year we do a national survey and there'll be a topic of interest and a survey sample selected. So last year we did sandwiches and ready-to-eat sandwiches and salads, and that report will be published in probably next year. And then also a lot of food businesses have agreements with their customers, so there will be testing done in accordance with that. And maybe Catherine probably would have more experience in this area. Yeah,
that's a very good point. From the retailer perspective, I suppose they've been slightly ahead of the curve with this. So a number of the retailers would have us even on our shelf life testing in triplicate, but with Listeria testing right through to the very, very end. One in particular would also, or two in particular, would call it a 20% over. So if we're giving X amount shelf life, same as the shelf life guidance would give, you have the 20% over, and we would be testing based on our commitment to them, Listeria right to the very end. So still working to the not detective in 25, but working it right through our shelf life to the very end.
Okay, perfect. And Catherine, this is for you. Do they do CCTV surveys on drains annually? Do any external specialists monitor the internal drains, or is it done in-house?
We do CCTV drains annually. That's quite new. We've only done that in the last 2 years across all the sites, and our swabbing schedules for our drains are done internally. We do a wide range. They are done during production as well. So our drain swabs are always done during production. There are some different options if you were concerned about your drain, about things like drag swabs, where you leave the swab down for the duration of the day to get a really good picture overall. But specifically for that question, yes, we do, but internal for our swabs, which are then sent to the lab for testing.
Perfect. Thank you, Catherine. Another question for yourself. Oh, and it just moved on my screen. Do you take L-mono environmental samples of a product contact surface and areas very close to the product contact surfaces during production?
Yes, all our samples, all our Listeria swabs are taken during production. So whether that's food contact, non-food contact, and drain. Yeah.
Okay, and someone is asking if we could revisit the explanation of absence in 25 grams versus less than 100 CFUs.
Okay, in terms of the method, well, I suppose,
They haven't.
okay, so there's two different criteria for that category 1.2, and one is the 100 CFU per gram, and that is a quantitative test, and it's less sensitive. So that's where the food product is added to a diluent, and then that's plated onto a special agar, and the lab will count how many Listeria are in that particular product per 25 grams. For the detection method, a portion of the food is taken and put into a special buffer, and that will enrich that product, and it'll help any injured Listeria monocytogenes to grow and recover, also maybe Listeria species. And then it's a qualitative test, so it'll say if Listeria is detected in that food portion or not. And as Catherine had mentioned, I suppose Listeria monocytogenes is a Listeria species. So the test is for Listeria species. So it is important that your laboratory determines if that Listeria is monocytogenous or not. Only Listeria monocytogenes is considered to be pathogenic, and that's why only Listeria monocytogenes is featured in that microbiological criteria. Then in terms of deciding which criteria a food business needs to comply with, really is around if you have that evidence to show that Listeria monocytogenes, if it's in your product, will grow beyond 100 CFU or not. So if you can show it and you've that evidence is presented to your competent authority and they accept that evidence, you can use that 100 CFU per gram limit. And if not, you're defaulting back to that not detected in 25 grams limit. I think if that person goes to the EU guidance, it really explains it in a lot of detail, and there's a decision tree there as well around which criteria is more appropriate to use.
Okay, thank you, Mary. So someone wants to know what advice you would have for supplier control when raw material naturally can have Listeria monocytogenes present.
So I would say, I suppose, characterizing the risk and associated with those raw materials is important. If you have a material that you know is more likely to be contaminated with Listeria monocytogenes, one option could be to use that at the end of the production day. So, before, if you use it at the start of the production day, you're risking cross-contaminating anything that you make after that. If you use it at the end of the day, then you can do your full kind of clean down, and maybe you would also build it into your verification program for sampling. So you might be testing raw materials, maybe looking at your actual supplier. Is there certain suppliers that you're more likely to get that product contaminated than others? Yeah, I don't know. You probably have some other tips, Catherine. I suppose I don't work in the food industry, but
No, you're bang on. I think the other things you could possibly consider as well is what your plan is to what's your process for that product. So are you looking at a chlorination step? So do you have something that may assist in reducing that risk? Are you considering UV? Is there a lethality step? Are you cooking it? Is that going to reduce the risk? I think if you have something that's inherent and you don't have a process that can guarantee to remove it, absolutely. It's all about your timing then, about doing it last, making sure that you're not cross-contaminating the rest of the product that's going through that system. And then your sampling. Is positive release realistic? Can you do that? Would that help control and not release it then out to the public? I suppose it also comes back then to the whole piece around, can you prove that it's not going to grow while in the industry? So.
Okay, thank you. The next two questions are about whole genome sequencing. So if a site identifies a persistent environmental Listeria issue, should whole genome sequencing be used as part of root cause analysis approach? And then if it is, I suppose the next question is asking how would you communicate these findings to the competent authority?
So I think whole genome sequencing can be a really useful tool for a food business to understand the profile of the Listeria monocytogenes. There might be multiple strains circulating in their business if they have a persistent problem. So you can get strains that are persistent to the factory and maybe have been there over a long period of time, or you might get transient strains that maybe are coming in a raw material, and they're sort of appearing intermittently, for example. I think it can also really help the food business to tie in with their environmental monitoring program, and as part of their root cause analysis, to kind of understand what strains are particularly associated with maybe, different parts of the operation, maybe in terms of equipment or drains, for example. Or sometimes what happens is a food business might find Listeria monocytogenes in a drain and then it's in a product, and um, they jump to the conclusion that it's come from the drain, but actually whole genome sequencing can show that the strain in the product is different to the one in the drain, and actually there's another strain that's coming from somewhere else. So it can really assist the food business to understand the profile. It is, in terms of communicating that with the competent authority, I suppose if you find Listeria monocytogenes in your product and it's a breach, you're obliged to inform the competent authority anyway. But I think the competent authorities, I mean, a lot of food businesses we've worked with and with the National Reference Lab, it's becoming more commonplace to use environmental or to use whole genome sequencing to assist investigations. And I would just see it as another tool that can really help food businesses understand what's going on. Do you want
Okay.
to add anything, Catherine?
I think everything that you said,
Yeah.
we have used it in the past, and I think that piece is absolutely accurate about the red herring, where people assume the drain is the natural source, so it must be the same as what's in the product. And we found exactly that when we dug into it, it wasn't. And I think I suppose from the competent authority piece as well, depending on whether you're, under the HC or under the Department of Ag, it's that working closely with your assigned person, whether that be a veterinary inspector or an EHO. It has to be reported anyway. There has been some work done with Chagas in the past as well about that sequencing of products that naturally occur in, say, something like a potato salad or in veg. So that there is a good bit of work behind it as well, which put with your own sequencing could give you a very good guide as to what you're finding. But it's an excellent tool, and will very much rule in or rule out a source as well.
Okay, thank you, Catherine. Someone's asking about control of Listeria in the cleaning process, and is there any particular types of products anyone would recommend? I assume there is resources available on various websites on this, but maybe you want to answer if you have.
Yeah, so I'm assuming they mean products being the cleaning agents or the cleaning products.
I think
Yeah.
we're talking particular cleaning
Um,
products, yeah.
so depending on what supplier you use, I think in current environment, most chemical suppliers will help you really build that robust cleaning program in the background. Some will even give you templates for how to validate your cleaning. From a Listeria specific perspective, I would say in a high risk environment, you'd want to be fogging anyway, whether you're just using a standard sanitizer. If you have an issue, something like a peracetic acid based chemical, a fogging chemical, where I can get right in there, and then chlorine
Yeah.
tabs in your drains and things like that. But honestly, the chemical suppliers are best advice and do provide excellent advice for control of Listeria mono in the environment.
Okay, thanks, Catherine. Someone's asking about the implications for the retail sector on the new EU regulations on El Mono.
Sorry, can you repeat that? Sorry, Marit.
Yeah, I'll just find it. It's already been answered according to our. Yeah, what are the implications for the retail sector of the new EU regulations?
The retail sector, well, I suppose, I mean, I think a lot of retailers already had this more stringent criteria in place in their customer
Yeah.
specifications anyway. Um, I suppose working with their suppliers to to make sure that they're prepared for this legislation change. I have been talking to FSAI have a retail forum. I presented at that. I'm presenting at the arts or the food service forum on Thursday. Um, I guess, for retailers to understand the requirements, and I suppose, I don't know what's going to happen, but we may have some non-compliance if a more sensitive test is used, and some food businesses not ready for that. But I think the best thing is really to be working with their suppliers and making sure they're doing everything that they can do to control Listeria in their business.
Okay, perfect. Thanks, Mary. There is quite a few more questions. If you're happy to stay on, everyone. Yeah, quite a few. So this is a few questions in one. How do the new regs apply to the the ready-to-reheat product that is sold solely to other FBOs, which would have their own FSMS question mark? Is that
So currently a product that is ready to reheat, that has full cooking instructions, as considered to be a non-ready to heat product,
yeah.
is not part of it doesn't come under the regulation 2073 microbiological criteria. But that said, there is an obligation on every food business to understand the risks associated with the food that they produce and to produce safe food. And we've seen from last year's outbreak that, for some of these products, even though they were labeled to be fully cooked or fully reheated, we did have some issues, and we ended up having 9 cases, and unfortunately one of those was a fatality. So I suppose to understand the risk profile again for your product, if you're selling business to business, I would say the risk is lower in terms of that food business that you sell to should have, as I mentioned, food safety management system that they follow in terms of making sure that product is fully reheated. And if Listeria monocytogenes will be destroyed if a product is fully reheated. But really, for businesses that are producing those non-ready to eat, ready to heat type ready meals, I would recommend they go to guidance note 46, which is our new guidance note, that gives advice to food businesses around these products, and understand if there's a reasonably foreseeable risk in terms of Listeria monocytogenes, should it be determined as a hazard in their process and controlled appropriately. And really, in terms of reasonably foreseeable hazard, I mean in terms of is there reasonable evidence to show that maybe some consumers might choose to not fully reheat those products and may eat them maybe warmed up for palatability purposes, which will not kill the Listeria monocytogenes. Or maybe if you have a product that has a serving suggestion, eat hot or cold, that's a ready to eat product because a consumer can eat that without heating.
Okay, thank you, Mary. Someone's asking, what pathogens should a small FBO be testing their products? Sorry, it's moved again.
Okay.
Yeah, sorry,
Yeah.
I'll go again. What pathogens should a small FBO be testing their products for shelf life testing? How can we advise? Can the laboratory advise? Is there any guidance on this that we can direct the FBO to?
Yeah. Okay. So
Yeah.
I think it depends case by case, depends on what the product is that you're making. The guidance we have is guidance note 18. Food Safety Authority Ireland's website, you'll get that. And that's the guidance note on how to approach producing shelf or determining shelf life for your product in general, not just specific to Listeria monocytogenes. Um, we also did a webinar with the Safe Food about 2 years ago now, maybe, right? And
Yeah.
that also gives a lot of information about how to determine what pathogens to test for, what
Yeah,
hygiene indicators.
that's still available on our
Yeah.
website. Yeah. So
Yeah, so really it's like understanding your product, what are what are the legal requirements, what are maybe this national best practice, guidance note 3, if it's right to eat food or not, um, what are the pathogens that are maybe associated with that product, so it really depends on the product and kind of looking at it sort of from that view.
Perfect. Thanks, Mary. Someone's asking about rapid tests. Are there any in-house rapid tests that are valid methods of detecting Listeria, or is it only external laboratory methods and retaining products until results come back for the products
Okay,
that can't be controlled by pH and water activity?
right. So there are some rapid methods, but you'd have to risk assess whether that is suitable. You might not want to have a pathogen on your premises that you culture up out of a sample. There's a risk there, I suppose, in terms of maybe cross-contaminating your products. So a lot of food businesses don't have their own lab on site, or if they do, they're testing for maybe hygiene indicators and they're sending the pathogen testing to an external site, or they have an external lab. In terms of rapid tests, food businesses and laboratories can use those once they're shown to be equivalent to the reference method, so the ISO method that's listed in the legislation. I don't know, do you want to add anything, Catherine, from a food industry point of view on that?
Yeah, so there, I suppose in the last few months we've been approached by a number of people with these like rapid methods. So, a pink blue reaction within 1 or 2 hours to a swab, a surface swab within the site. They're lovely and they're great if you maybe had an issue, if you had a prevalence and you wanted to try and keep on top of it and just find directions going. I wouldn't say that they could be replacement for your lab testing in any shape or form. The other side of that rapid testing, exactly as you said, if it's equivalent, a lot of the rapid methods are not. And you may get negatives where the approved method would be a positive because it's slightly more accurate. It's one to be careful of. Labs can give you very good advice as well, but from a rapid method, we've only ever used it as a top up if there was an issue or something like that, not on its own.
Okay, thank you, Catherine and Mary. Is the 2-day safety margin applied by many FBOs to shelf life enough? The durability studies suggest 7+ assessment. So,
I don't think I fully understand that question, but
yeah.
I think in terms of a margin of error, in Guidance Note 18, we would recommend food businesses maybe take back their shelf life by a day or so, considering maybe some consumers might choose not to abide by that use by date. But it's very case by case as well, and up to the food business, depending as well on the shelf life. Like if you have a 7-day shelf life, taking it back by 2 days is quite substantial. So you'd have to have a rationale for that. It's definitely not something that's required, but depending on the product, you may choose to do it as sort of an additional safety measure. But I think it depends on the business. Catherine, do you have any feelings on that, or?
I find that a lot of the retailers will lead in this one as well. So some require rather than a 2-day, a 20%. So I suppose if your shelf life is short, it probably will land in around 1 or 2 days. But the risk is absolutely that your customer or consumer will not abide by that and look at it and go, "Ah, looks fine, keep going." So we would always build in at an absolute minimum a 20% margin of error there.
Okay, thanks, Catherine. Someone asking about swabbing methods. Do you use the pyramid method, i.e. swab most floors and drains? I assume that's directed to yourself, Catherine.
Um, We don't use a pyramid method per se. In our high risk area, I suppose it's worth noting as well that we're a very divided area. So each line has its own zone, for want of a better description. So it's built over years of data. And we do tend to move our location, but no, we would try and cover all drains over a period of time. And then depending on if we're getting positives or negatives, we would increase that frequency. All drains absolutely would be covered, but I think to get a happy balance between food contact and non-food contact is just as critical as hitting a drain. And not being afraid to swab places of concern. So again, behind those slicer blades where if the blade isn't taken off, you may not have gotten a proper hygiene clean. Behind panels, that's a biggie. Those panels don't come off in a general hygiene wash down. It's not, it's just take those swabs and go everywhere, really. So and then based on your results, you risk assess your frequency.
Okay, the next question is about swabbing as well. So when carrying out Listeria swabbing, is it advisable to swab 2 or 3 areas using the same swab, or should every individual place be swabbed individually? We currently have a risk assessment to justify swabbing more than one area with the same swab. Is this acceptable in practice, though?
So I suppose I can only speak from experience here. We have found that the labs tend not to stand over composite swabbing.
Okay.
So if you are putting, say, a number of swabs into a bag, any recent feedback I've had from labs is that they won't stand over that as being a test method that they can validate. They're not validated for a composite swab. If you're wanting to cover a large area, let's just say, for example, you had floor damage in multiple areas, to narrow that down or to try and get a picture of what you're doing, yes, you could use that swab in, say, 3 or 4 different flooring areas. Your risk there, obviously, is if it's in one, you're going to cross-contaminate to another. So it depends on your risk assessment, depends on your environment. Yes, it's possible, but it's very much it's case by case and how you risk assess that out for yourself.
Okay, thank you. There's
Maybe
still lots
just,
of questions
sorry,
to get through. Oh,
just
sorry,
just to
Mary.
add there, Mairead, I suppose I would
Yeah.
see a disadvantage of maybe swabbing a number of areas, not to mention maybe possibly cross-contaminating another area, is when you get that result and it's positive, you've no idea which location that positive actually is corresponds to. So it's probably in one way more cost effective, but actually in the long run, it's slowing you down in determining the root cause.
Okay, the next question is, what corrective actions would you advise for the detection of Listeria inocia or Welchemere in product or the environment?
So I suppose we would advise that food businesses would take similar corrective actions, proportional, I suppose, to the risk. If it's a ready-to-eat food, we wouldn't be looking for, to notify the competent authority in terms of a recall or anything because it's not pathogenic. But it's a hygiene indicator, and Listeria monocytogenes occupies the same ecological niche as other Listeria species. So if you're finding Listeria species, particularly in your product, that means possibly Listeria monocytogenes could also cross-contaminate that product. So I would investigate it in a similar manner.
Okay, thanks, Mary. Okay, the next question, what does good look like for a scientific justification for a product if there is Listeria detected in a 1.2 group product, externally validated pH and internally acceptable overlife, preservative levels in recipe? Sorry, Mary Ju.
Well, I think if you're finding Listeria monocytogenes in your product, it's too late to be justifying that it won't grow beyond 100 CFU per gram. You should be doing the groundwork before that, and coming to your competent authority with your evidence. And if the competent authority assesses your evidence and determines that actually if Listeria monocytogenes gets in that product, it won't grow beyond 100 CFU per gram, the competent authority will make a note for that environment or for that FBO that the limit that they can comply with is 100 CFU per gram. But if you haven't done all that homework and you find Listeria monocytogenes in your product, it's too late maybe to be doing some work after that fact to find the pH, etc. I think you need to sort of do that work beforehand and have that as part of your food safety management system.
Okay. Catherine, did you want to come in there?
Just I suppose from their estimate scientific evidence, Mary mentioned it in her slides as well. Every product is different. So a handmade in one site can vary so much from a handmade in another site. So even that scientific literature that's there that may guide you into believing that your product will not grow, I think you need to be very careful about your considerations. Every site is different, every process is different. So it would need that homework done, even if there are similar products that have scientific literature behind them. I think you need to be more robust than just literature on its own. I think that's
Okay, thank you, Catherine. Okay, the next question is, for a medium-sized food business cooking their own hams and beef for retail from their own premises, who have documented their understanding of their product, have their own HACCP system and CCPs in relation to cooking their own meats. They have their own sampling plan under CAN3. Also, they do environmental sampling and have undertaken shelf life studies, but do not undertake any 2073 testing. What is the best place to start in tackling compliance with regulation 2073?
Okay. I'm not sure what they mean by not doing any 2073 testing. So it sounds from that type of product, there are criteria that they need to comply with. So I suppose looking at the legislation, determining which criteria you need to comply with. Now, for a lot of the ready-to-eat products, there's no sampling frequency in the legislation, so you base that on a sort of a risk assessment. I'd say if you're starting from scratch to understand the requirements of 2073, I would recommend going to FSAI Guidance Note 27. And that's our guidance note on how to comply with Regulation 2073. And there are some complementary e-learnings on that in the FCI e-learning portal. You'll find them there.
Okay, thank you, Mary. Someone's asking, would you recommend testing Listeria species in 25 grams over Listeria monocytogenes in final product?
So 25 grams is the amount of sample that's taken for the test, whether it's in final product or, at the end of manufacturing. The standard ISO actually is for Listeria species. So the lab will be testing for Listeria species, and depending on your laboratory, they might report that differently. The laboratory should also always confirm if there is Listeria species found in that product, what it is in terms of if it's monocytogenes or not. Some laboratories might report it as Listeria species innocua. They might give you that species information as well, or some might just report it Listeria species. But as I mentioned, if Listeria species, it's recommended to include that as sort of a hygiene indicator. So if you find Listeria species in your environment or in your product, it's showing you that possibly the conditions also exist to that Listeria monocytogenes could be there, and you take the actions in a similar way. I don't know, do you want to add anything, Catherine?
No, that's exactly it.
Okay, perfect. Someone's asking, can there be too much focus on drains as a possible source of Listeria rather than a collection point?
Definitely. I think we tend to look down and don't look up. And I think that's really important that you look at everywhere on your site. Chiller units, fans, things, AHU units, they can be just as much a harbour point for Listeria as your drains can. So yeah, and I think again with that new guidance note, that gives you a much clearer picture of this whole thing around the non-food contact and food contact. Even things like stopper buttons, e-stops, under panels, you got to go digging, I guess, because if it gets in, it stays in. And drains can give you a false sense of security. If your drains are coming up negative, you can kind of walk away feeling happy, and it could be in your chiller units or something like that, especially when you think of perhaps a product coming out of an oven into a blast where it's steam and it's condensation that could sit in the roof or sit in the chillers and be circulated around. So we need to look up and out as much as we look down, for sure.
Okay, thank you, Catherine. Someone's
Jade,
asking,
sorry to interrupt. Just before you go on with any questions, we're coming up to our time when we said we were planning on ending, but the webinar is so interactive and we have so many questions. Our presenters had agreed to stay on, and that's brilliant. But what I think we might do is say we'll go on for another half an hour until 12, and then we'll call it at that stage. And if there are any questions we haven't been able to get to, we'll ask our presenters if they'd be happy to answer them separately, and we hope to then include those answers with the link to the recording, which we'll share. I hope you're all okay with that. Catherine, Mary, Kevin, if you're okay to stay on till 12. Yep. Brilliant. Great. Sorry for the interruption.
No, you're fine. Thanks, Tracy. So someone's asking, are you essentially expecting scientific justification for product in 1.2 group or historical and risk-assessed environmental, raw material, barrier performance going to make a difference, including cook and other decontamination? Irrespective of all controls, does group 1.2 need to have pH, preservative, water activity, or max 4 days life?
I think yes. I think this whole talk has covered that. So, as part of controlling Listeria monocytogenes is having all that food safety management system in place. But also you need to think about your product, and you need to show if Listeria is there, how it will grow.
Okay, thank you, Mary. Someone's asking, has anyone any experience of using enzyme cleaning chemicals?
So yes, we would mostly use them in our slaughter plants. They are very good. Some chemical suppliers steer away from them. I wouldn't have any experience of them specifically in controlling of Listeria mono, but from a hygiene perspective, they're excellent. They are certainly an advantage because the gross cleaning and rinsing is reduced. So the enzyme cleaner, you just need to do a quick clean and apply the chemical, whereas standard chemical, you'd need to do a gross clean, a rinse, a chemical application. So from that perspective, it can maybe save time, but I haven't seen any literature to date on specific control for L mono around it.
Okay, Catherine. Someone's saying they currently use chlorine tablets in our high-risk drains. Should we follow suit with our low-risk drains?
We use them in both, just to be sure. And again, it's down to that understanding of which way your drains go. We do a level of low-risk Listeria swabbing as well, which would be not the norm, but again, it's for our own health space. We've got that raw material coming in, particularly, I think there was a question earlier about something that maybe has a level of or natural occurring Listeria in it. That low-risk control becomes just as important. So yeah, a low-risk swabber would, maybe not at the same frequency as high risk, but it would be a good idea.
Okay, thank you, Catherine. Someone's asking, is how to reduce the risk of Listeria in the environment and hence in the product? And also, what is the acceptable as reliable scientific justification if Listeria is detected in 1.2 group? Will the historical controls and general controls in the environment, barriers, etc., suffice as scientific justification in the event of detection in 1.2 group?
So no, I suppose is the answer really there. I mean, we would be looking at the pH, the water activity, the preservatives, etc. But we'd also take into consideration, your general approach to how you manage Listeria monocytogenes in that business. So I think that information is more for the food business to determine their own risk. And a lot of that is covered in Guidance Note 45.
Okay, thanks, Mary. Someone's asking, in addition to more sensitive methods, will there be more testing done by authorities?
No, they're not planning on doing more testing. So at the moment, I don't know off the top of my head how much testing is carried out, but the testing amount will remain the same. The only difference will be that it's going to switch for those 1.2 products to the detection method.
Okay, thanks, Mary. Someone's asking, can you confirm if within Chapter 1, sampling plan N equals 5, does that apply to a food business operator or the competent authority?
The food business operator. So that's in Article 1 of Regulation 2073. That Regulation 2073 specifically applies to the food business operator, but the competent authority can also use it when assessing compliance or verifying compliance of food businesses.
Okay, someone's asking, but can we send the link to the webinar that we talked about a few minutes ago? And yes, we can. We can actually share that link as well with this webinar link if people would like that, no problem. Okay, someone's asking, do you consider that consistently negative monitoring results over the years for Listeria are possible?
If you've got good control, yeah, it's possible not to find Listeria monocytogenes in your product or in your environment, but I would say it's ubiquitous, and I would look a bit more closely, particularly at the environmental monitoring. If you're consistently finding negative results, is your environmental monitoring program robust enough? Are you testing the places that you think it might be there? And I suppose the problem with negative results is that sometimes they can give a false sense of security. And you need to remember with Listeria monocytogenes and all bacteria, they're not distributed uniformly. So you could, within a batch of product, you could have a low level of contamination that's sporadic throughout the batch. That's why the more testing you do, the more likely you are to find it both in the environment and in the product. But of course, it's a balance because the more testing you do, the more expensive it is. And I think you need to have a risk-based approach to your sampling program. And again, Guidance Note 45 really talks about that. Do you want to add anything there, Catherine, around negative results?
Yeah, I think I suppose it's a little bit like back to the focus on drain. So if you're getting a full year of absolute negative results, I do think you need to go back and great if it is, but I think it's where you go back and look at things like your floors. Is there any points where there's pooling water? or maybe a damaged floor or damaged flashing where something can get in down behind it. So while the drain is getting naturally cleaned and like that, maybe you're using chlorine tablets and you're keeping it at bay, there may be other points throughout your process areas where you were actually finding harbourage, things like your wheels on your trolley, which is in your guidance note 45, things like that. It's yes, the more you test, the more you find. And I think people may be slightly afraid of that they'd rather just stick with the status quo where if I'm testing these 12 drains, I don't find it. I think we need to look at it. It's better to find and eliminate than not know it's there.
Okay, thanks, Catherine. Someone's asking what action is required if you get Listeria monocytogenes on a contact swab?
I presume they mean a food contact surface. So you would risk assess the result. And if it is possible that food has been contaminated, you need to take that into consideration. And maybe if you have some end product testing, you might have the Listeria monocytogenes also detected in that end product. So it needs to be on a risk basis. And again, guidance note 45 really talks about that and what you do in that situation.
Okay, thanks, Mary. Someone's asking, as a cheese cutting facility for wholesale and retail, is not detected in 25 grams test at the end of the shelf life enough to justify a 25-day shelf life? Should the whole wheel cheese producer detail the new changes in their product specs?
So I think for any product, you need to assess what the shelf life is suitable for your product. And I think for cheese, I suppose you're looking at if Listeria monocytogenes gets into your product, how will it grow? And if you're finding it would grow to beyond 100 CFU per gram, you're in that not detecting 25 grams. In terms of the shelf life, the 25 days, I suppose, is made up of maybe the organoleptic profile, the hygiene profile. There's a few other things you need to take into consideration. Would that product spoil over that time? But in terms of Listeria monocytogenes, for that business, it's really about making sure that Listeria monocytogenes doesn't get into their product. So if it gets into their product and Listeria is detected at any stage during the shelf life, it will be a non-compliance.
Okay, thank you, Mary. Catherine, someone's asking if you can explain in a bit more how to use the lag phase in ComBase or other models to capture the WIP life of ingredients before they go into finished product, if I understood correctly during the presentation. Thank you, if that's possible.
Yeah, no worries. So I actually wasn't talking about the lag phase, just the actual product
Okay.
life, but I do understand where they're coming from. In ComBase specifically, there is little information buttons that will give you a more detailed understanding of each of those areas. So for the pH or even your starting point where there's giving you a base log of Listeria, which you can slide back and forth and put it to 0, or you can have a starting point of maybe 3 or 8 or 10 CFUs or whatever you want to do. But specific to your product, if you click into those information buttons for your lag phases, that will help. But again, if you're considering worst phase, you've got to go right back to the start. So your oldest raw material, your meat, if you have open dry ingredients, how long they're opened, and consider it the whole way through.
I might just add to that around lag phase specifically, we would recommend that 1 is used as the default, which is the worst case scenario. Basically, lag phase is around how quickly the Listeria monocytogenes would start growing in your product. And 1 is that it would start growing straight away. If you had some justification to show that there is a different lag phase value that you could use for your product, you would need to have proof and evidence for that. But as a worst case scenario, it's recommended to use 1 as your lag phase.
Perfect, thanks, Mary. Someone's asking, is excess steam an issue for some food business operators to argue that it is not condensation?
I think you'd have to, not sounding funny, you'd have to prove it. So yes, excess steam can be. So if you imagine pulling your racks or your trolleys out of your oven and the steam hits, while the steam may not be condensation, it is condensing on your roof. So anything that is on your roof that may be there, perhaps Listeria, can then cross-contaminate with your steam. Condensation control, steam control, anything like that is absolutely critical to your Listeria control. So if you have sufficient extraction post-cook, as your ovens are open, especially if you're using steam ovens, from a retailer perspective, they would consider steam condensation one and the same. Once it hits that cold barrier, hits the roof, it is condensing.
Okay.
So you have to have a very robust assessment to prove otherwise, I think.
Thanks, Catherine. Someone's asking, what are the panel's views on the use of phage technology, such as PhageGuard, as part of cleaning and disinfection protocol?
So I suppose in terms of some of those products, they are not permitted for use in the European Union. There was one brand in particular that I know has been going through the courts about that. But in general, the reason I suppose the European Union see that under Regulation 852, which is the one that contains HACCP and prerequisite requirements, that good hygiene and good food safety management systems is critical in producing safe food. And that businesses shouldn't be turning to decontaminants to remove contamination at the end of maybe manufacturing, for example, that it shouldn't be there in the first place from manufacturing it in a hygienic, under hygienic procedures. So at the moment, that those types of products are not permitted for use in the EU.
Okay, thank you, Mary. And we're on to our last question. So this one is about water activity. We currently accept the result of 0.97, not over it, for water activity in our meat products. However, I seen on one of your slides equals than or less than the result of 0.96. So they're obviously asking, is that acceptable?
That was just an example, I suppose, from one of our products. So we would, depending on the product, we may have water activities ranging from 0.95 up to 0.98. So it was just for demo purposes.
It was just for Perfect. Okay. I'm conscious, Kevin, that you never got to say anything. So if you'd like to say anything now before we close the session, in particular.
So that was easy money, wasn't it, Maria? No, it's probably just worth saying, and I know we do still get queries about it, whether or not the amendment applies in Northern Ireland. So, just in case people aren't aware that, of course, the UK, which Northern Ireland's a part of, has left the EU, but following that, due to the movement of goods between Northern Ireland and the Republic of Ireland, they put what was in place called the Northern Ireland Protocol, which kept Northern Ireland aligned with certain EU rules so that goods could move freely between the north and the south. That caused a bit of complications politically in Northern Ireland. So then in February 2023, they reached the agreement, which is called the Windsor Framework Agreement, which replaced the protocol. And in simpler terms, it was just designed to make trade smoother so that EU law would still continue to apply. And Northern Ireland was to fix the practical and political problems that were about scrapping the protocol altogether. So the requirement to adopt 2073/2005 is contained within Regulation 852/2004. 852/2004 is listed in Annex 2 of the Windsor Framework Agreement and therefore is directly applicable in Northern Ireland. So that's where it comes to the point where 2073/2005, along with the amendment, is still applicable in Northern Ireland. Just to clear that up in case there happens to be any confusions among FBOs either in the North or in the Republic.
Perfect. Thanks very much, Kevin. That's brilliant. And just, I suppose, to finish off, thank you so much again to Mary, Catherine and Kevin for joining us all today and all the participants. We'll be sending out a short feedback survey to you all shortly, and we'd really appreciate if you take a few moments to complete that. We will have the recording up on our website shortly with some of the useful resources, the links to those that Mary shared in her slides already. And I suppose if you'd like to hear more about our upcoming events and you're not already a member of our knowledge network, you can sign up at safefood.net. And just again, thanks for joining us today and hope everyone has a great day. And Tracy, is there anything else I should have said that you'd like to mention?
Just to remind people, if they wouldn't mind completing the short survey we send, it's very useful, very informative to ourselves. So we'd be grateful for that. And that's it from us. Thanks to everyone again.
Thank you. Thanks for asking us. Bye.
Thanks, guys. Bye-bye.